Welcome to the Jack Westin MCAT Podcast with your hosts, Michael Maleka
and Molly Hudash. Hello, everyone, and welcome back to another episode of the Jack Westin MCAT Podcast with your hosts, Mike and Molly. Mike, I'm excited to talk about our topic for today. Coming out of last week's episode, we were very focused on genetics and Mendelian inheritance, so genotypes and how they influence your phenotypes, right? How the two different copies you have of each gene influence whatever characteristic they encode. But we kind of left out a lot, which is how that actually happens, right? Like, how does the set of alleles you have for a gene, how does that create a phenotype, right? We talk about DNA as like, oh, DNA codes for everything, but it literally does not. It codes for everything, but it itself is just the instructions to make important things. So today we get to talk about how DNA actually controls the phenotype. Super, super cool.
So interesting, right? This idea, and I think this idea is going to be really, really central to the whole conversation, is that every single one of your cells has the exact same blueprint. Every single one. Again, maybe I should say most, like 99% of cells have the exact same blueprint. But yet, our cells are so different, right? So therefore, there must be some process. It's not just like copy paste, like DNA. Okay, now that's just what the cell is. There must be some process for how we actually take that information from the blueprint, take what we actually need, and then create the functional properties, which is going to be proteins. And this process from DNA, that blueprint, to protein is what we call the central dogma. The central dogma is kind of like the trait of like genomics. It's kind of like not the trait, the saying, or like the poster boy of genomics, where it's like
a dogma.
It's like the central dogma. Like if I do the actual term, where it's like you go from DNA to RNA to protein. That is the central thing. Of course, there are some there are some exceptions to this. You know, sometimes you don't go all the way to protein. Sometimes you can go backwards. But like the central idea in genetics, the central dogma is that you start at DNA, you create RNA from it, and then you create protein. And that's going to be the focus of today, is the process of how we go from DNA to RNA. That process, then from RNA to protein, what's involved in that. So that's what going to be our real focus of today, is that process of the central dogma.
Yes. And this is one of my favorite things to talk about. So we're going to literally talk about DNA to RNA. What does that look like? It's a process called transcription. We'll talk about it. We have RNA, which then gets made into proteins. And like you said, there are some times that RNA is the final product, and cool, we love that. There are examples, we will give them. RNA to protein, that's translation. But one of my favorite things to talk about that's even one step deeper with what we talked about last week, we're not just going to talk about how this happens mechanistically. We're going to talk about when it happens. Because like you said, every cell in your body, for the most part, has the exact same DNA. And yet, you do not see the same physical characteristics or enzyme expression, right? In many of our cells, they have unique functions, right? And that comes with a different pattern of gene expression, right? So we'll talk about actually how we take DNA and make RNA, but we're also going to talk about when this will happen for particular genes, and what cell types will express certain types of genes, and how one cell type could be specialized to detect light coming into your eye, whereas another one can be specialized to, sense changes in your blood glucose and secrete hormones, right? Like gene expression, right, is the process by which we take DNA and create a functional product. But we're also going to talk about gene expression regulation. So how we control this process, because not every cell expresses every gene. And especially, this is not even true given, timelines, right? Like you might have a cell express a certain gene early on in development and not later. So we'll talk all about that as well. Super cool.
Super cool.
This is one of my favorite topics because
it's so interesting. Right? It's like the foundation of everything we study. Like in anatomy, like this is like where it all begins. We're also going to touch on not only how like that regulation happens, but throughout we're also going to distinguish like eukaryotes, which are what we are, and prokaryotes, things like bacteria, because there are key differences the MCAT will expect us to know about these processes. But I think those also cool. But before we like dive into like all like this process of transcription translation, I think it's important that we know like the key players in this kind of game and this kind of like process that we're going to talk about. So some key players to note that we're going to bring up repeatedly today. First is DNA. Like what we need to know what DNA is. DNA is this anti-parallel double helix, and I want you to think of it as the blueprint of the human body, of the human. Right here, it's all of your genomic information coded in A's, C's, T's, and G's. All of your coding is 4 letters. This is actually kind of going back to like when they first like kind of discovered DNA. DNA also has protein involved in it. They're wrapped around these things called histones. People thought when we first like started like figuring out what codes a human, they thought it was the proteins, that because proteins have like 20 different amino acids, they're like, oh, humans are so complex, right? Oh, it must be all of those 20 different amino acids, the sequence of those, that's what codes it. But in reality, no, it's just the 4 different like bases made up in your DNA. That's this anti-parallel helix, and this is kind of like where we start off in terms of the human code. Then we have to talk about like what reads that code. Like how do we go from this blueprint? Oh, you want to mention something?
Just in case anyone's hearing anti-parallel double helix and being scared, it's just 2 strands of nucleotides. So, right, sugar, phosphate, and a nitrogenous base, like you said. There's 2 of them, and they're wrapped around each other, and they're going in opposite directions. That's it. But if anyone's hearing that, it's just 2 like, oh my goodness, chains of nucleotides, and we're describing how it's arranged in space.
Absolutely. I have always heard that term. I'm like, yeah, everybody knows, but you're totally right. We should really explain what that means. But thank you for that. But we have to talk about, okay, we have this 2 strands of nucleic information that's anti-parallel, but that on its own can't really do anything. Again, it's the blueprint, but we need to first read the blueprint. And that's in order to turn it into that first step, which is, RNA. And that's the job of RNA polymerase. That's a key player. We're going to go into more detail about that process in a little bit. But when we bring up RNA polymerase, it's important to know that is kind of the enzyme, the protein that actually reads the DNA and turns it into RNA. But you mentioned before, there's RNA is sometimes the thing that goes to protein, but sometimes RNA is the final product. So we know there's different types of RNA that we can see kind of in the body. And there's kind of like 3 major ones. There's like some minor ones that we'll talk about a little bit later on, but there's kind of like 3 major types of RNA, and that's mRNA or messenger RNA, tRNA, and there's rRNA. So these are kind of like these RNAs are kind of similar to DNA, where DNA is deoxyribonucleic acid. RNA is ribonucleic acid. It doesn't have the deoxy, but we have these different types of RNA which denote different functions, and we're going to talk about those a little bit later on. Then we have ribosomes, which are kind of the next kind of key kind of protein enzyme involved. There's not just protein, there's also RNA involved in this thing, which ribosomes are what take the specifically mRNA, not tRNA or rRNA, and turn it and read it and create a protein from that code. Again, I hesitate to say turn because it's not like it transformed the mRNA into a protein. No, it simply reads it and it uses that as like the instruction for the protein. So we'll talk about ribosomes as well as transcription factors. Transcription factors are these things that key regulators when it comes to, when we, like you said, we don't always express all genes all at once. Transcription factors are things that we're going to mention that tell our body when we need to express certain genes at what times, or things that always inhibit certain genes for different cells. So those transcription factors, when we bring up those, kind of regulate when we transcribe things and when we don't transcribe things. Ooh, that was a long rant. That was a long ramble.
We're going to talk more about every single one of these things. These are just the key players. Again, we're talking about transcription and translation, and then we'll dive off the deep end with gene expression regulation, which would be fun. But again, in terms of biomolecules, we're primarily focused on nucleic acids, so DNA and RNA, different types of RNA, yes, pretty much one kind of DNA. And then proteins, right? RNA and proteins can have weird 3-dimensional structures. We'll talk about why. DNA, though, always are double helix. Okay, so now we've kind of set our baseline. Let's move into our first step of our central dogma. Now, oftentimes when you have a central dogma lesson, you might start with replication. But I want to be clear, replication is not truly gene expression. Replication occurs, let's say, before a cell divides from 1 into 2. And it's just, hey, I'm about to go from 1 cell to 2. I need to make sure that each of those 2 product cells, or daughter cells as we call them, has DNA. So I'm going to replicate my genome So I now have 2 copies of my genome so that each cell gets one. That has nothing to do with gene expression. Replication is important, especially when we talk about things like the cell cycle, mitosis, meiosis, all that good stuff, and with mutations and cancer and things like that. But it's not part of our central dogma in the truest sense of how DNA is made into proteins. Okay, so we're going to start with transcription, which is how we take DNA and we convert it to RNA. Now, one of the reasons I bring in replication is because oftentimes that's what people start with. And also, replication has so many enzymes. Okay, it can feel a little overwhelming. You're talking about topoisomerase and helicase and single-stranded binding proteins and DNA pol 1 and 3. And there's a leading strand and a lagging strand, and it's very overwhelming. RNA polymerase in transcription really is like, for the purposes of the MCAT, that's your enzyme.
That's the start of the show. That's the
start of the show. It does it all, right? It's like a little multifunctional enzyme that really just runs the show. Okay, so if you have RNA polymerase attached to a particular strand of DNA, it can just go. Okay, it's only interacting with one strand because one thing that I don't know if we officially mentioned, RNA is single-stranded. DNA is double-stranded, RNA is single-stranded. So we are going to find DNA.
That for anyone watching, she just pointed. For anyone who's not watching, in the background there's a DNA double helix. That would be a great example. If I brought, why didn't we bring that up when we talk about anyway? But yeah, that Molly just always has. But that's a great visual.
Yeah. So ultimately, RNA polymerase is going to identify which strand it wants to use to make a new RNA sequence of nucleotides from the DNA. And once it gets started, it's going to go. Okay, so let's break this into our 3 steps. We're going to use these same 3-step names for translation as well. Okay, we say initiation, which is kind of getting set up, getting started. We have elongation, and we have termination. Okay, so it's like beginning, it's the setup, actually doing it, and then how we decide how to end, right? Initiation, I want to be super intentional. This will only happen when conditions are right? And we'll talk about regulation of transcription a lot, okay? But assuming conditions are right, what we're going to see is RNA polymerase, our, multifunctional enzyme, is going to bind to a sequence in the DNA called the promoter. Okay, and this is what is ultimately attracting RNA polymerase. We'll talk about how the conditions are right in a later segment. RNA polymerase is going to bind the promoter, and we are going to begin, right? That's really the setup. There's not a ton of context to provide other than we have 2 strands of DNA. We have 2 strands of DNA, and one will be used as what we call the template strand. The template strand serves as the template for transcription. Okay, so we're going to take one of those 2 DNA strands, and I'm going to synthesize an mRNA that is complementary to that template strand. Okay, and it's going to be the same. We talked about DNA being anti-parallel, so moving in opposite directions in that double helix. It's the same thing. Okay, so I'm going to pick one strand that has that sequence RNA polymerase will recognize, and then I'm going to basically unwind it and create a new mRNA. We call that the template strand because it's used as the template. The other strand of DNA is called the coding strand, and there's something interesting going on with that other strand. When we think of double helix structure, those 2 strands are running in opposite directions, but if you have a C on one strand, the G will be on the other side. If you have an A, you'll see a T on the other side. They're complementary. If we take the template strand and we synthesize mRNA that is also complementary and also anti-parallel, right, moving in opposite directions, what we see is the coding strand has the exact same sequence as the mRNA will, with one exception. mRNA, apart from the fact this is ribonucleic acid, it's a different sugar, it also has U instead of T, uracil instead of thymine. So other than that, right, the coding strand of the DNA will look exactly the same as the mRNA. They have the same sequence because they're both complementary and anti-parallel to the template strand. Know that RNA polymerase figures this out. RNA polymerase recognizes the promoter on the strand that it likes, and it's going to be able to, during elongation, which we'll talk about, separate those two strands apart and actually just start synthesizing RNA. Right. So once we figure out what to bind to,
it's going.
We're pretty
it's
much
gone.
good.
Yeah, it's gonna go. That's the initiation, and we kind of touched on elongation a little bit, but it says during this elongation step where it's like going along that kind of segment of DNA, reading that segment of DNA, specifically in the 3 to 5 prime direction. It reads it in the 3 prime to 5 prime direction because we can only synthesize nucleic acid in the 5 prime to 3 prime direction. And because, like you said, they're anti-parallel, they're going in opposite directions. If I can only make 5 prime to 3 prime, then I have to read it 3 prime to 5 prime. And so as it's exciting down, is creating this mRNA sequence, yes, that like is nearly identical to the coding strand. That's why we call it the coding strand, because it kind of codes for it, even though that's not what we're actually reading. And so during that elongation step, it's just going. We are really making it really simple by saying there's only RNA polymerase involved. Like we're like, because for the MCAT, that's what you need to know. Yeah. In reality, of course, there are other enzymes involved. Of course, reality is more complicated. But for the MCAT, all you need to know is RNA polymerase keeps on chugging along, keeps on elongating, creates this mRNA. But we know mRNAs are not all the whole length of the DNA. So we know eventually we need to begin termination. Now, outside of the MCAT, there's all of these sorts of factors that are involved in termination, like the forming a loop and that sort of thing. But all you really need to know is that there's this terminator region of the DNA that once the RNA polymerase reaches the terminator region, it's simply going to release the mRNA. Right? Once that RNA polymerase reaches it, it's like, okay, I'm done. Of course, I'm simplifying for the purposes of the MCAT, but then it's going to release the mRNA. But one thing that's very, very, very important, right, is then what? Because I've said something that I need to really distinguish between prokaryotes and eukaryotes. If we were talking about prokaryotes, this is mRNA that we're releasing, right? But in eukaryotes, it's not really mRNA yet, right? In eukaryotes, we call this hnRNA. Because in prokaryotes, because there's no nucleus, this is done, right? It's no nucleus, so as soon as transcription's happening, it's getting translated. Because the nucleus separates like the ribosomes normally, which we'll talk about, from the nucleic acid itself. Because in prokaryotes, there is no nucleus. That's the whole thing with prokaryotes. They don't have organelles or membrane-bound organelles. As soon as that mRNA is released, a ribosome's grabbing onto it and starting translation. Right. And so in that way, there's no processing that can even happen in a prokaryote. But in a eukaryote, just because we've created that strand of RNA doesn't mean we're done with it. In a eukaryote, it needs to be processed before we can actually consider it mRNA. And so this processing, and then after it's processed, that's when it can actually leave the nucleus and actually get translators. We'll talk about this a little bit. The processing, there's kind of a few factors involved. First, before it can actually become an mRNA, this hnRNA needs to be spliced. And what we mean by splicing is all of those nucleotides that were kind of copied from the DNA, not all of them actually are part of the protein that we want. This is where we hear the terminologies introns and exons. Because introns are the region of the hnRNA, really, they're not part of the mRNA. They're part of the hnRNA that need to be removed, right? Introns get removed, exons stay there, they remain. And so I was always confused by this because I was like, oh, introns stay in, and exons, exit. The exact opposite. It's the exact opposite. Introns are what exit, and exons are what stay in. That was a mistake I always made all the time. But I mean, maybe you have another thought.
I mean, I think it's more introns stay in the nucleus, and exons exit the nucleus. And they get to leave.
That's a better. Because I was always confused, like, oh, introns should stay in, but no, that's a better way. Introns stay in the nucleus.
Introns stay in the nucleus because they're spliced out.
Right.
Exons stay in the mRNA, but they get to exit the nucleus. Yeah.
That's a great way to think about it. See, I never thought about it this way. As I was just like, whatever I think it is, opposite. But that's an even better way to think about it. All right. So that's one of again, this is specifically in eukaryotes that the splicing happens. Again, splicing, not a prokaryotic thing. As soon as it gets transcribed, it's translated right away. But another thing that happens in terms of eukaryotes, in terms of processing, is the cap. we add a little hat on the top of the 5' end of our mRNA, which is known as the 5' methylguanosine cap. And all you need to know about this cap, it's on the 5' end, and it serves one of its main function. Two kind of main functions of this 5' cap is one, to stop it from getting broken down, but two, it helps with recognition by the ribosomes later on. The final processing we need to do for our future mRNA is on the 3' end. We need to add what we call the poly A tail. Because like we said before, the RNA is going to have A, C's, G's, and U's, right? That's going to be all that what actually makes up the mRNA. But if we just we had the 5' cap and left this kind of string of nucleotides, as soon as it exits the nucleus, we've got all these little exonucleases, right, in the cytoplasm, that if we don't do anything is just going to chew up our nucleic acid. So what we do is we actually add this kind of string of A's, just a whole bunch of A's at the end of the nucleic acid. That's why we call it the poly A tail. A whole bunch of A's, so that when it gets released, they start chewing on the A's, because the A's don't really code for anything. They're just kind of like a way to abide the exonucleases until we can actually get what we need from the mRNA. Again, I've really kind of gone into detail about the modifications we do to mRNA, but kind of in summary, specifically in eukaryotes, after this kind of hnRNA is released from the RNA polymerase, we splice out the introns, we add a 5' methylguanosine cap, and we add a poly A tail.
Yes. And I feel like it's interesting to mention the differences, right? I feel like DNA, I always visualize as a total germaphobe. I will not leave my house. Okay. I am staying here. DNA
Right.
should not leave the nucleus, right? It's always protected. mRNA is being created from DNA. It's a list of essentially what DNA wants, right? DNA's wishes, right? But ultimately, in order to leave the nucleus, it needs protection. It needs armor. The reason is our RNA is not meant to last forever. DNA is supposed to last the entire duration of that cell's life, because we only really replicate when we're ready to divide. You need to protect your DNA. RNA, though, you want to have a shelf life, so to speak. You don't want that around forever, because if you are prompted to express a gene at a certain point, we'll talk about regulation in a moment, you want to express it. You want to create that RNA. You want to make sure that it's protected for the duration that you need it, but you don't want to keep expressing that forever. Right? You want each RNA to have a particular period of time that it can stay alive, so to speak, and stay active, and then you want it to go away. So the protections are a temporary, it's temporarily giving that RNA the ability to survive in the cytoplasm, but ultimately, like you said, that poly A tail is going to be chewed up, right? This RNA is not going to last forever, and it shouldn't, because that's what allows us to respond to changes in our environment. So very, very cool. And just so you know, when we talked about splicing, right, introns stay in the nucleus, exons can exit the nucleus, right? Those are what we'll talk about. We'll actually code for protein. That being said, you actually, it's possible if you had a gene, let's say, that had 10 introns and 11 exons. Cool. You could cut out those 10 introns and all 11 exons stay in there and they all leave the nucleus, and you make one protein that's encoded by 11 exons. That's totally fine. There's also a way that you could take that transcript, cut out all 10 of the introns, and also, let's say, 2 of the exons. You don't need every exon, so you can have one gene actually create multiple proteins in that case, right? If you only let 9 of the 11 possible exons leave, right? Now your protein that would be translated from that will have the amino acids encoded by 9 exons instead of 11. We typically call these isoforms, right? We form these through what's called alternative splicing. Right. And I feel like it's worth mentioning RNA, those sequences that are introns. So many students are like, why do we even have those? Are they just junk? No, they're not junk. There's a lot of very important regulatory sequences like splice sites, which are recognized by proteins that ultimately remove certain parts of your RNA. you said hnRNA, which is totally true. Also, sometimes they'll call it pre-mRNA.
Sure.
Because it's RNA and it's going to become mRNA, but it's not quite there yet. We haven't finished processing.
Right.
So
and I think the idea of alternative splicing, I'm sorry, I have to mention it because I think I remember when I was first learning this, I always thought the bigger the genome, the more complex the organism. Because you would think, oh, more genes, more complex. But there are like plants, like ferns that have longer genomes, like that have like
by a lot,
by a ton. But then it's like, okay, how, and like even like simple organisms with way bigger genomes than humans, but obviously we are more complex than a plant. I mean, I'd hope so.
Whoa!
Yeah, I'm gonna
plant hate off our podcast.
I'm gonna go out on a limb, and that more humans are more Hey, debate me in the comments. Let me know in the comments if you agree. But a major explanation for how we can be so complex with a smaller genome is alternative splicing, that we can get multiple different proteins from the same gene, from the same region of DNA, which I think is a really cool idea.
Yeah. Plants are really cool. I will say they actually can duplicate their entire genome as a way to become different species. True. It's really cool. It's very cool. Plants are awesome. We are not plant haters.
No plant. Hey, no plant.
We are human. Here. We are human biologists in the lens that we are preparing you for the MCAT. But you know what? No plant hate. But yes, and ultimately, right? This brings us to translation. Okay, because at the end of our transcription, we have a pre-mRNA or an hnRNA we need to process because, again, prokaryotes don't need this. We do. We have to leave the nucleus, and you do need protections. We are now out in the cytoplasm, and we're going to begin associating with things like a ribosome, right? Binding or having things like a tRNA come and bind us, and we're ready to get started. Let's talk a second, though, about what an mRNA looks like, a mature mRNA, right? Because this is also going to relate to our other types of RNA as well. Okay, so ultimately, we've got a long sequence of nucleic acids. These are ribonucleic acids. So ribose is the sugar. We are going to see our first amino acid that will be added is going to be added with respect to what's called a start codon. Okay, that's when we are actually going to begin. There is a exact sequence, and you should know this. It's 5' AUG 3'. So that's the directionality. AUG codes for the first amino acid. That said, there's a whole segment of that mRNA before and after that is not going to be translated. We call them untranslated regions or UTRs. These typically have roles in regulation. Okay, again, there's never blank, boring, useless DNA, right? Or RNA in this case, right? Like they typically have roles. Do they get all the attention? No, not really. But we're going to see the start codon. AUG will always code for methionine. That will always be the first amino acid added. And then after that, we're going to see 3 nucleotides read at a time in a given reading frame, which is what we call it. So it's a long sequence. We read the first 3 nucleotides are AUG. And then the next 3 nucleotides could be anything. It could be AAA, it could be GGG, it could be CAC, it doesn't matter. And you read them 3 at a time. Okay, when we talk about mutations, sometimes we mess with that reading frame, right? There's a lot we could talk about there, but you read 3 at a time, and we call those 3 codons because each triplicate, right, or each triplet, I guess I should say, codes for 1 amino acid, okay? And so these codons are going to match up with what we call anticodons on tRNA. tRNA is what's actually going to bring the amino acid to this translation complex. So tRNA is going to interact with mRNA and ultimately will facilitate the elongation of our protein. Okay, so I just want to make sure we're all comfortable with what an mRNA looks like because we will always start at AUG. Always. And when we talk about the actual mechanism of translation, that's going to become really important, right? The other major player, right? We've got tRNA, which has a very fun, like almost 4-leaf clover looking structure.
Shape, yeah.
tRNA is going to be charged. That's the word we use for it. It's because this process that requires ATP, but it's charged with an amino acid. It will physically carry it. Okay. And again, it has a sequence that can form hydrogen bonds, right? Complementary base pairing with that mRNA. Okay. So they're complementary, just like DNA and DNA or DNA and RNA in transcription. Same process. So complementary and anti-parallel. And then ribosomes. This is a complex of both rRNA, right? So ribosomal RNA and protein. So I want you to notice, like, as far as our key players, we see all of the major types of RNA at play here. We have a mature mRNA. We see tRNA, which is going to bring, it's called transfer RNA, brings the amino acid close. And we see our rRNA, which is ribosomal RNA. Okay, this complex that we call the ribosome is ultimately going to house translation, right? There's even two subunits, a top and a bottom, a large and a small, right? That ultimately clamp onto the mRNA and that will move around to facilitate the elongation. So this is the actual, think of it as like the warehouse in which this takes place. Within that, we have 3 sites, right? We call it the ape. You say ape.
I like
say
ape.
EPA.
I always like ape. I don't know.
It's fun. I mean, ape is more fun because it is, we'll talk about initiation, elongation in a sec, and termination. Oftentimes it's written as E, P, and A because of essentially
how it goes down,
how it moves forward. Yeah, but A is the attachment site, P is the peptide site, which ultimately holds the growing chain, right? Peptide site has the growing chain of amino acids, but then that will be moved, we'll see this in a sec, to the peptide or the tRNA in the A site, which is the attachment site. And then we'll have the E site, which is ejection. Okay. So I always did EPA. I think it's because I love the environment. I think of the Environmental Protection Agency.
I didn't even
it just comes up.
I didn't even clock that you were like saying the EPA. I was like ape. Monkey. Yeah.
Yeah. But also it's usually written EPA if you look at like a diagram of translation. So that's even more. Yeah. But technically ape is a good way too. So these are the different actual sites within the ribosome, right? So we know the structures. I think we're ready to get into the actual steps.
Translation.
Initiation, elongation, and termination. It's the same exact thing. I feel like there's a lot going on in all 3 of these. Before, we kind of just said RNA polymerase does it all. And as long as you show RNA polymerase to where to go, it just does it. It does it all, right? This is not the same. So there's a lot of steps. We'll go through each one by one.
Right. And so I think the first step is start, like imagine we've just released that mRNA from the nucleus. Like mRNA with this little cap on, it's ready to go. It's got this little cap, little hat on. It's now left the nucleus. And now it needs to get started. But we have to do something a little bit first. So you mentioned before that ribosomes have 2 subunits, the large subunit and the small subunit. What's important to know is that they're not always connected. Like before we've actually started, they're actually separate from one another. So before any translation begins, the small ribosomal subunit, the small one, actually is already bound to the specific tRNA that will recognize the start codon, the AUG. And I don't think this is something that we mentioned, but every tRNA, there's a separate tRNA for each amino acid. So there's a tRNA for methionine, a tRNA for alanine, there's a tRNA for, they're all matched together. So the tRNA for methionine is already bound to the small ribosomal subunit before any mRNAs even entered the picture. So that's just floating around. But once that small ribosomal subunit with that tRNA for methionine meets the mRNA, it is now going to bind to that mRNA and recognize and find where that start codon that you mentioned was, where that AUG was. And so because that tRNA from methionine has the anticodon for it, it's going to go and recognize where that AUG is. So it's going to hold on there. And once it's found, oh, hey, I found the start codon. My tRNA is there, start codon's there, small ribosomal subunit is there. That's when it says, okay, big ribosomal subunit, can you come? We're ready to get started now. So the large ribosomal subunit is going to go bind to the small one, and now you have your whole ribosome complex. But what's very, very important, I see this kind of idea all the time, is where does it actually start? Which site, A, P, or E, of the large subunit does that first tRNA begin? And a lot of people think, oh, it's the A site because that's the like start site because A is first. No, it starts at the P site, right? Where that large ribosomal subunit is going to come, bind to the small one, and that tRNA for methionines is already at the P site. It doesn't need to start at the A site. It's already at the P, and now we have begun, we've initiated, and we're ready for the second stage, which is elongation.
Yes.
Yeah.
And so, like you're hitting at, people are like, oh, it should go into A. A is for attachment, right? Or some people will say it's for amino acid, like singular amino acid. Either way, whichever you want to use works, right? And it's because in elongation, that's exactly what's going to happen. Okay, so coming out of initiation, you are basically at the first stage of elongation. You have something in your P site, okay, and that's it. Now, the first portion of elongation, which is ultimately, right, starting right after the start codon, right, has been sort of surrounded by its tRNA and its rRNA. The first thing that's going to happen is you're going to have another charged tRNA enter the A site, which you could think of as the attachment site or amino acid because it's individual. Okay. The correct tRNA anticodon that will bind to the codon that's in the reading frame, right? Note that like you always go 3 at a time. And it's because you have, your AUG is already bound to something. So you have to go to the next 3, right? Ultimately, we enter the A site, right? And then we take the whole peptide, which if we're talking about the first step of elongation is just the methionine, but could also be a growing chain of a very long list of protein or of amino acids in a protein chain. That chain is going to be added to the amino acid in the A site, right? Because it's the attachment site. We take the P, the peptide, from the P site, and we move it to the amino acid in the A site. Okay, now what we've got, nothing in our E site, which is the ejection site. The P, I have a tRNA, and yet it's not attached to anything anymore. It's uncharged. There's no amino acid attached to it. And then I've got a tRNA in my A site that's got a super long chain of amino acids, hypothetically. At least it's got more than one, okay? So that's where our protein chain is now living, so to speak. Now, most of the time when we talk about what happens next, we think of all of the individual tRNAs shifting over, let's say to the left if the ribosome is moving to the right. But really what we see is the ribosome can sense that my growing chain is now in the A site, and the ribosome just scooches 3 nucleotides forward. Okay, so ultimately what was in the A site is now in the P site. The tRNA is not moving. The ribosome is moving. So what was in the A site is now in the P site because the ribosome has moved forward, right? And the tRNA that used to hold the peptide is now in the E site. E is for ejection. We basically toss that thing out.
It's
empty now.
Useless. It's gone. We don't need it anymore.
And then we just rinse and repeat. Another amino acid or another tRNA carrying amino acid that matches the codon with its anticodon will come in, right? We will have the transfer of that peptide to the end of that amino acid. The ribosome will move forward. We now have one in the E site and the P site. Get rid of the E, join in the A, and so on. This is the vast majority of translation, right? This takes a long time, but we slowly do this. And I want to just in this process, we are reading so far, and we're not talking about replication today, but so far, whenever we do nucleotide to nucleotide, we read 3 to 5 and we synthesize 5 to 3. In this case, we are reading 5 to 3 and synthesizing from N to C. N is the amino terminus, C is the carboxy terminus. So 5 to 3, N to 3, or 5 to 3, N to C, is the directionality. So we are slowly reading from the 5' at the beginning to the 3' end, and we are starting with the N, right? The N terminus is where methionine is always going to be. It's the first one added, and we are going to keep adding to the C terminus. Right.
Keeping on going, rinsing and repeating, APE, all the way down until, and that's the kind of where we get to kind of the third phase. First, we start with initiation, then elongation, and now we have to terminate because our proteins, we don't want to make them infinitely long, obviously. So, but there needs to be some communication, something that the ribosome reads that says, hey, we need to terminate. And that's where we get to the stop codons. Now, for the MCAT, there are 3 stop codons. Another term I've seen is like an amber codon is another term that I've seen before. So there's kind of 3 stop codons you should know of. And we both have like different mnemonics for this. Mine's not so friendly. Yours is a lot more friendly. So I kind of wanted to hear yours first. But I think it's better to hear yours first before you hear mine, about how to remember what the 3 stop codons are.
To be fair, I think we actually have the same one that we learned. This one, that's nice. I actually heard from a student. Oh. I told my mean one to the student, and they were like, oh, haven't you heard this? So it's UGA, UAG, and UAA. So it's you are awesome, you are great, you get A's. Aww. Isn't that sweet? I heard that and I was like, that is so much nicer than how I always remembered it.
Right. Because that's the nice way, not nice way that I learned is you are annoying, you go away, you are gone. So UAA.
Mine's even meaner. You are gross. You are annoying. You go away. That's the way I learned it.
Oh, you're actually Oh, wow.
Which is actually one step meaner than yours. But again, the mean stuff sticks. So
the mean stuff does stick. That's true. That is very true. But yeah, those are
the weird stuff sticks, I should say.
So
because some of the silly ones stick too.
Yeah. But to go over, because I know I went into mnemonic. Yeah, UAA, UGA, UAG, those are your 3 stop codons. And what those do is when it reaches there, it's essentially to say like, hey, there's no really tRNA for it. It's more just, hey, stop, let go, right? We have now finished. We are now done. Again, I want to point out this is over towards the 3'. This is at the 3' end of the coding region. It does not mean it's the end of the mRNA. Like you mentioned before, there's still the untranslated region after the stop codon, and then maybe there's a poly A tail left that hasn't been chewed off yet. But yeah, this is important to know that after this point, now the whole complex kind of just, again, for thematic purposes, blows up. It doesn't actually blow up, but it's just it just comes apart at that point, where now it actually releases the mRNA and that growing peptide chain. The actual release of the peptide chain does require ATP. But now, hey, now we've created our protein. Amazing, love that win, and we have our mRNA.
Yes. So we're done, right? And when we think of the central dogma, again, to kind of reflect and pause for a moment, most sequences go all the way through from DNA to protein. That's the vast majority, because protein really is the workhorse of our body, right? Like we need sugars, we need fats, we need lots of other stuff. True, yes. And we need DNA and nucleotides, but really in terms of structural roles, in terms of enzymatic roles, in terms of like transport proteins, like proteins are so diverse. They're so unique, right? They really do the majority of the work in the body, right? And ultimately, they are informed by DNA. That's how we create proteins, right? As we've talked about. But there are some times, like in the case of rRNA and tRNA, and we'll talk about some other very minor types of RNA as well, that we don't go all the way through, okay? And that's fine too, right? But ultimately, this is the way that we get all the way through to proteins. Now, when we talk about the differences here between prokaryotes and eukaryotes, again, in prokaryotes, we have no membrane-bound nucleus, right? There's no nucleus at all, which means, and this is super weird and awesome, as we are transcribing, right? Again, we are synthesizing from 5 to 3, right? We start from the 5 and we keep adding to the 3' end. What's really cool is even as we're synthesizing, as we are transcribing, translation can be happening. Like literally, like you don't even have to transcribe everything and then it's translated. Like you could literally be transcribing and translating at the same exact time. That's why we have no introns, exons. That's why we don't need a cap or a tail. Like there's no alternative splicing. Prokaryotes, there's zero separation. What is transcribed, even if it's an ongoing process, can be translated. That's a major difference, of course, and we need to know that for the central dogma. A lot of regulation is very different, and we'll talk about that in a moment. The other thing that's really commonly tested that you do want to know is the difference in ribosomal size.
Yeah.
And this is actually a target of a lot of antibiotics. So it comes up a lot in passages. And it's also something that's super weird. So in terms of size, right, we talk about it like size. It's actually using Svedberg units. I don't want you to worry about this. It's basically how these proteins sediment during centrifugation. So if you spin it up really fast, it's describing basically when it begins to come out of a liquid solution. I want you to know that this is not like, oh, this is mass. And the reason why is it doesn't add up very well. But what I want you to know is the small and large subunit of the ribosome are different sizes from ours in Svedberg units. And the total size is also different. So what I want you to know with prokaryotes is they're all the odd ones, right? They're odd. So 30S, and again, S is Svedberg. 30S is the small subunit. 50S is the large subunit. And this is where it gets weird. You'd think 30 plus 50 is 80. It should be 80S. It's not. It's 70. Again, it's because it's not a unit of mass. It's a sedimentation rate. Don't worry about it. 30, 50, 70. Small unit or small subunit, large subunit, and then the whole complex. We are a little bit larger, but we're all even. So we're 40, 60, 80. So I also would know that. If they're talking about 80S ribosomes, that's us. If it's 70S ribosomes, that's a prokaryote. Yeah. And again, we have ways that we can target these different types of ribosomes. We could deactivate them. And now, if you are a prokaryote using prokaryotic ribosomes, you can no longer translate. If you take away the workhorse of a bacterium, it's not going to be able to create disease as well. It's not going to be able to set up shop and create an infection to the same degree. And that's how it can be used as an antibiotic, which is pretty cool.
Which is cool. I also want to note, by us, we mean eukaryotes, but because it's like
that's true.
30, 50, 70 for prokaryotes, 40, 60, 80 for eukaryotes, including us, as
including
we are
us.
Eukaryotes. I didn't want to make anyone seem like, oh, it's just humans. Oh, but other eukaryotes are just other things. Yeah. But yeah, all eukaryotes have this 40, 60, 80. So it includes like yeast cells, for example. Personally, I love yeast. That was where my research was in yeast.
You love yeast and you hate plants? Come on.
Well, it's actually a fun guy. But I think this also brings us to, like, we've gone through the central dogma. We've talked about how we go from DNA to RNA. We've talked about RNA to protein. We've talked about differences between eukaryotes and prokaryotes. But now I think it's important to talk about what we mentioned kind of at the beginning of today, which is about regulation. Because we know you've got a whole bunch of genes on your DNA. Not all of your cells express all of your genes. That would mean every single cell would be identical. And we know, just from being humans, that's not the case. We have, photoreceptors. We've got nociceptors. We've got neurons. We've got skin cells. We've got all these different cells, but they all essentially have the exact same DNA. So there must be a difference as to what they actually express. So we're going to be talking about different levels of expression because we know humans are so complex. So we obviously don't have just one expression, like expression regulation methodology. We have so many kind of levels where we control expression, both pre-transcription, post-transcription, pre-translation, post-translation. We've got all these places that we can regulate so that we can finally tune and control what genes are expressed, not only where, but when as well, which I think is really, really cool.
Yeah. I love this. It's so, so cool. I imagine it's like, imagine you're trying to go drive a mile on like a city road, right? In the middle of New York City. Like you hit stoplights at so many different places. There are so many places where you have to pause and you can have regulation occur. You could have someone directing traffic or whatever, right? That's what we're saying here. So we could have a stoplight before transcription even happens and say, no, you know what? Don't transcribe this, right? You could have it directly with transcription. Again, like, will this be transcribed? Yes or no? You could have it after transcription. You could have it after it's been fully transcribed. It's left the nucleus. It's been processed, all that good stuff. But before it's translated, you can have after it's translated. There's so many contexts. Okay, so let's start. So cool. Ultimately, the vast majority of our gene expression regulation is going to be on the level of transcription. Okay, so when we talk about whether something is going to be transcribed, we have to first ask, is the DNA accessible? And then we have to ask, is the DNA attractive enough to RNA polymerase to begin? Okay, so those are the first two that we'll talk about. The first thing is DNA even accessible? Like, could RNA fit if it wanted to? That's when we need to talk about the structure of DNA. And I don't mean, anti-parallel double helix, right? What I mean is we've got a long strand of nucleotides, right? A full chromosome. Typically, this DNA strand is not just left, right? Think of it like a thread. Have you ever seen like a spool of thread? If you just let that thread go with no spool, right, it would be a mess. It would get tangled, it would get damaged, you would pull on it, and it would get, it's a nightmare. Okay, so we have proteins that act like the spool that a thread is wound around, right? We call these our histone proteins, okay? And DNA, when it's wrapped around it, is called a nucleosome. Okay, so we have these proteins. I want you to recognize, though, if you have like a spool of thread, that's not going to be very easy to access, right? So if you want to use and express a particular gene, you have to unwind it from the spool. You have to make the DNA come off that histone in order for RNA polymerase to physically be able to fit. Now, we could talk a lot about exactly why DNA is wrapped around. I'll give you a nutshell just because it's cool. The histones contain a lot of lysine residues. Lysine is a basic amino acid that is positively charged at physiological pH. DNA has a sugar phosphate backbone, and phosphates are negatively charged. DNA loves those positively charged histones, right? The negative charge on DNA and the positive charge on the protein, they want to wrap up, okay? If we go in and disrupt those positively charged lysines and make them neutral through a process called histone acetylation, we now have DNA that's negatively charged wrapped around a histone that's neutral. It's not so enticed by that. DNA is going to fall off because it's no longer held to the histone. Histone acetylation, which neutralizes that charge, allows DNA to come off, which ultimately can facilitate or accelerate transcription, right? There are also different ways that we could talk about epigenetics, and these all are epigenetics, right? Non-heritable or non-structural changes in DNA that could be passed on between generations, but that are not due to actual changes in nucleotides, right? We could also talk about DNA methylation. You can methylate certain nucleotides that ultimately silence them. But this is a major point of regulation, right? And we call DNA in its unwound form, right, chromatin. When DNA is wrapped around really, really tight, we call that heterochromatin. Okay, hetero means different, and it actually, when you were looking at giant protein DNA complexes versus small segments of DNA that loop between them, looks different, right? Because you have proteins in there. When DNA comes off, right, we call it euchromatin. And I love this. It's EU chromatin. I love this because euchromatin is used. Euchromatin can be used.
I've never
right?
heard that before.
Heterochromatin
Cool.
cannot be.
Yeah.
Yeah. So if you have DNA tightly wound around those histones, that's heterochromatin. If it loosens up, again, in that case, it is now physically possible for RNA polymerase to come over and find the promoter. Once we find the promoter, RNA polymerase is happy. But we have that second situation. It needs to be physically possible for RNA polymerase to bind, but then even calling back to our initial discussion of transcription, conditions have to be right. RNA polymerase must want to bind to the promoter. I always like to think of it like, we've established RNA polymerase does so many jobs in transcription. Like, I like to think of RNA polymerase as constantly buzzing around and having a million different genes it's supposed to work on and transcribe. It needs not just access, it needs something to wave it over and say, hey, prioritize me. I know you're busy, but this gene is ready to be regulated, or it's ready to be expressed, right? And that's when we get into a whole lot of other fun things.
Right.
Transcription factors.
Transcription
I can't
factors, activators. And this is where, when we were talking about before about like, RNA polymerase, that's the sole like thing that does this thing. If you actually looked at like an image of like RNA polymerase transcribing, you'll see the RNA polymerase. It's the core, but then you'll see all of these like little balls, again, by a stylized image, like attached to it to kind of tell the RNA polymerase where to go. That's where transcription factors come in. Because like you said before, most of the regulation happens pre-transcription, or like to tell what to transcribe, because it's a lot less efficient. Like if you already did the work to transcribe it to regulate it after, most of it happens before transcription, or to at the transcriptional level. So like you said, the promoter is there, but a lot of times the promoters, while available to the RNA polymerase, isn't actually so sticky for the RNA polymerase. It doesn't actually can't grab so tight. So what will happen, depending on certain environmental factors, like let's say, you're stressed versus not stressed, or maybe you are hungry versus not hungry. Different environmental factors can influence it, different times of the cell. Transcriptional factors are going to go and actually bind to the RNA polymerase and kind of attract it to the promoter to say, hey, this promoter, I'm the transcription factor for this promoter. Like, hey, we're hungry, you should do this one right now. Or we're stressed, go transcribe that promoter over there. Again, the promoter was maybe always there, but it wasn't maybe attractive enough for the RNA polymerase. And that's where the transcription factor comes, binds to the RNA polymerase, binds to the promoter, some region next to the promoter, and says, hey, you're coming here, you're transcribing this thing. So a lot of times you need those transcription factors. It's not just RNA polymerase that's involved, but in order to actually get the thing going. We can also talk very briefly here about like operons. Operons are something where these are involved, where we can control like the lac operon for like lactose expression. How we're not just always like these bacteria, for example. Again, for note, operons are only in prokaryotes, really, not really in eukaryotes. Operons are kind of these transcriptional control regions where you only want to transcribe this region of DNA in a certain condition. The lac operon is if I need to a lactase enzyme, or if there's lactose present. Obviously, like glucose sometimes is better, so you don't always want the lactase enzyme. But the lac operon is a great example of when just because the promoter is available doesn't mean you're gonna be transcribing it. You also see the trip operon, the tryptophan operon there. They're a little bit different, but just want to mention those briefly as good examples of transcriptional regulation when it comes to expression.
Yeah, operons are super weird. And again, like you said, prokaryotes only. We're mentioning here because it's basically how prokaryotes decide whether to transcribe something. And keep in mind, in this little aside about prokaryotes, once you transcribe, you translate. There's no pausing, right? If you decide to go, you're going all the way through to proteins. So operons or prokaryotes have to be very careful about what they choose to transcribe, because once you do transcription, you do translation, literally sometimes at the same time. So the way that they do this, and a way that they keep their genomes nice and tight and small, because they're small, is they have multiple genes that are encoded by the same, or they are all transcribed into the same mRNA. So multiple genes on one mRNA, we call it a polycistronic system. A cistron, I guess, is like a individual gene. Polycistronic means you can have one mRNA transcript that includes multiple genes with multiple complete proteins. Okay, multiple start and stop codons, different peptides that are created. But we cluster them. So like if you have lactose, let me activate all of the genes involved in lactose digestion. If you have lactose, we will transcribe and translate every protein that we would need for lactose. They group them like that. We do not do this, right? We're bringing them in because this is prokaryotes, but we don't do it. There are different types of regulation. Definitely worth knowing. Lac and trip are the big ones that are tested. The question is, are they always on, or are they off until you tell them to turn on? We're not going to get into specifics there with trip and lap, but they're pretty fun, right? Once you already have the transcript, because everything that we've said thus far is, will you transcribe it? Okay. You could also be thinking, for eukaryotes only, we have alternative splicing. After the mRNA transcript, or the pre-mRNA, or the hnRNA, as you describe it, after that's done, before you ship it off to the nucleus, you can have alternative splicing. We could also call back to our RNA discussion. There are also other types of RNA that play a very important role in whether or not an existing, RNA present in the cytoplasm ready to be translated will actually be translated. So we have miRNAs, right? And we have siRNAs. siRNAs are more so used in a lab environment. miRNAs are actually pretty common within our own cells. But we have sequences right? That can ultimately, single-stranded mRNA sequences that can bind other mRNA sequences that are single-stranded, and they form double-stranded RNA, which should not happen, right? Keep in mind, during transcription, you have a DNA RNA kind of hybrid. That's fine. And it's only in the nucleus. There's really no appropriate situation to have double-stranded RNA in the cytoplasm, right? Like that shouldn't exist. And if it does, if you have one of those interfering or inhibiting RNAs come and bind to a particular mRNA, it's actually going to target it for degradation. It's going to silence that mRNA. So there are ways after an mRNA has already been fully processed and it's left the nucleus that you can actually silence it and prevent translation from ever happening. Okay. miRNAs and siRNAs are pretty important there. Plus, keep in mind, RNAs don't last forever. They're meant to be destroyed, right? If we don't do any of these things, it's going to be translated. But even after translation, you could cut that protein up, right? You could cut it into 2 pieces. Maybe both those pieces are functional. Maybe they're not. We could talk about adding ubiquitin tags. Ubiquitin would be a functional group, essentially, pretty large functional group. But you can tag your proteins with ubiquitin, and that's going to basically tell the cell, go into my cellular recycling bin. I'm done with you.
Trash that protein right over there.
Trash that protein. Yeah, it could be glycosylated. It could be attached to a lipid. It could be phosphorylated, right? We can change activity and structure a lot after translation. But for the most part, just to reiterate, most of our gene expression regulation happens before transcription has even started. And that's true of both us, which is eukaryotes, you're right, and our prokaryote counterparts, right? We have more options because we have more stoplights, right? They basically have one.
It's like, hey, you're going and you're going.
Yeah. Do I transcribe? Yes. Straight shot. No more stoplights, right? We have a lot more flexibility in, different layers that we could regulate gene expression. But again, this is what gives us all of our different cell types. It's also what makes a, 2-week-old child different from a, 70-year-old adult. Like patterns of gene expression control everything.
Right. Which is so
I love that we get to talk about it.
Right. Yeah. And again, the MCAT will also test on it heavily. A lot of the times, kind of questions that they're going to ask are going to be very much on which level. Like they're going to say, oh, we inhibited transcription. What do you expect to happen? Oh, we inhibited translation. What do we expect to happen to RNA or protein levels? Oh, we added this siRNA. What do we expect to happen to protein levels? The understanding these are important because those are the kinds of questions we ask. But like we always keep on calling back to, you can't memorize your way through that. We have to understand why, like the importance of each step and the importance of each component in order to be able to answer questions like these. And I think I'm really excited to kind of talk about the next thing we had kind of planned, because we've kind of hit on how you inherit what you inherit, like Mendelian inheritance. We've talked about it. Then we talked about, okay, you got the DNA that you got from your parents. How do you actually show that, express that DNA? But we know growth is a huge part, and our cells are constantly kind of dividing and replicating, some cells a lot more than others. How that process actually happens is actually the topic of our next episode, where we're going to really dive into replication, mutations, cell cycle, mitosis, meiosis, and cancer. And I really like this topic a lot because that was like the topic of my like thesis when I was in school, is like all about like cancer and cell cycle, and talked about yeast a lot. That's a big target for these sorts of studies. So I am so excited for next episode.
I'm excited too. My favorite thing in the world is watching people completely nerd out over something they're really passionate about. And I feel like that's us every episode. But I'm particularly excited. Not only is it a high yield topic, but it's your research. So I'm going to have a million questions. If you have questions for us or for Mike about his research, you let us know. If you have anything that you want to see us cover in these episodes or questions you want us to answer, you tell us, right? We've got a comment section, right? Whether you're watching this on YouTube or Spotify or Apple Podcasts or whatever, let us know. We are so excited to see you in the next one. We hope you enjoyed this one. And tune in next episode for